So the next speaker is Ted Meets. Ted Meets joined us also last year from Amsterdam, actually, and he'll be talking about real science. Please.
Okay, so good morning. So I'd like to talk today about some a new project that's very early, so very preliminary work, but mainly I'd like to spend most of the time motivating sort of the modeling approach that I'm taking, and also maybe introduce this sort of application domain to people in the audience. So when when working with cancer genomics data, you always keep in mind this sort of overarching research goal, which is precision medicine. And the idea is that every patient has a unique disease, even though they share a lot of characteristics with other patients, but it's unique for that individual. And what we really want to find is the causal mutations that are driving their disease. And in particular, we want to find all of the sort of causal mechanisms so that we get the whole story for any patient. And then given that we know which mutations are causal, we want to know what are the downstream phenotypic effects of these mutations so that we can then intervene at some point with medicine. So what are the implications? So if you know which genes to inspect for mutations, then what you can do is you can sort of target your sequencing. And so instead of sequencing the entire genome of a tumor biopsy, you can really focus in on a subset of the genes, and even this can save a lot of money and is more feasible in a sort of clinical setting, even though the costs of sequencing are going down quite a bit. The other sort of more the more important implication for the patient is that we can improve the targeting medicine for this patient. So when we know the whole story for a patient, and we know that the sort of the effects of these sort of mutations on the patient, then we can give really targeted medical treatment for this patient. So we can sort of, instead of focusing on just on one mutation, which can kill the cancer, but then it can come back, you want to focus on all the possible mechanisms and have this sort of synergistic effect. And of course, if you understand all of these combined effects, then you can improve your drug development. So there's there's a vast amount of modeling challenges with this data. I'm only going to speak about the sort of the mutation data that's available. Okay, so one thing is this publicly available data from the Cancer Genome Atlas that I'm using. It's very rich, so it has a lot of things that are measured. Genotype, RNA, microRNA, methylation data. But it's a very small data set from what we're sort of used to doing in machine learning. So there's about 10,000 data cases in the entire database, and for any given cohort or cancer type, there's anywhere from only 80 to maybe 1,500 cases. And the data itself is extremely sparse in terms of the mutations per gene. The distribution of frequencies of the genes, which I'll get to, is extremely heavy-tailed. And it's also noisy, so there's a lot of false positives and false negatives in the data. But one of the main modeling, one of the aspects that I'm trying to focus on with the modeling is that you can have different mutations that have the same kind of phenotypic effect, so the same kind of causal mechanism. So some low-frequency mutation and a high-frequency mutation, they can have the same underlying effect, and we sort of want to combine that into a latent variable or a disease state across the cancer cohorts. So the modeling idea is very simple. So it's just a latent variable model of the RNA expression. And we're going to assume that these latent variables represent some kind of disease state. And you can think in the back of your mind that these sort of could represent disruptions to cellular pathways or circuits. And what sort of an important assumption to make is that we're going to assume that these disease states can be recognized from the RNA, and that also the disease states are caused by mutations in the DNA. So this is sort of where the reconciliation has to come in. So we want these disease states to be caused by the mutations, but also able to reconstruct or be also recognized by the RNA. And the sort of the distinction in this work is that we're trying to learn this a special kind of structured prior that's going to sort of combine sort of possibly mutually exclusive mutations in the data set to influence different factors. So we want to learn which genes combine together to turn on some disease factor. And we're going to do this by constraining how genes can influence a latent variable. So that's the modeling thing that I'm going to go back to later on. Just some overview of the sort of the data now. So cancer can be understood as a genetic disease, and that the mutations are the causal mechanism. So here you have an example of a normal cell. It gains some mutation, and then that will give it some kind of sort of evolutionary advantage within the body, and then that cell will then proliferate. And these sort of advantages that it gains can be either a gain of function, so it can start generating its own growth stimulation, or it can lose some function that sort of checkpoints in the cell cycle, or losing the ability to suppress tumors. We're going to assume that these effects can be observed in the RNA, which is we're going to sort of collectively call the phenotype for this talk. And then the progression of cancer sort of follows this accumulation of mutations. So as you accumulate different kinds of mutations, you gain these other kind of characteristics, or what can be called hallmarks of cancer. So you start developing genomic instability, and then at some point the cells can invade the nearby tissues, or they'll start stimulating for growth of blood vessels and so on. Now, underneath that picture is this canonical view of the circuitry that's going on inside your cell. So you have these different circuits that are important for the normal function of your cell. And in some of them, in cancer, some of these circuits have some disruption, and this disruption is caused by some mutation. So here's a mutation so it's a mutation in the DNA that can affect the protein that gets made by that gene. And so perhaps that protein doesn't bind to the other proteins in its pathway, for example. And so what happens is you have this downstream effect that can be recognized in the gene expression data. And what's important to note here is that you can have different mutations in different genes in the same circuit that are all going to have the same downstream effect. So you can imagine that you have a set of patients that have the same RNA profile, but they've had very different genes that have been mutated. And somehow you want to know which genes you should be sequencing so you don't miss that rare gene for that patient. So what does the data look like? It's very has a lot of interesting properties that make it difficult to work with. So along the x-axis here, I have the different cohorts for 20 different cancer types. So this one is colon cancer, for example. And the y-axis is the log mutation counts for each patient. So a circle here is the number of mutations that one patient has. And the two things you can see is that as you move across to the different cancer types, that you change about 1 or 2 orders of magnitude in the average number of mutations per patient. And then also within each cohort, the variation is quite strong. So you can have 1 or 2 orders of magnitude change within just one cancer type. Now, if you zoom in a little bit more and go at the gene level, and here along the x-axis you have different genes, and you can now look at the frequencies of mutations overall pan-cancer and also within the different cohorts. So these are the frequencies of the mutations, and then the colored by the relative frequency for different cohorts. So there's 2 things to point out here. One is that there's huge variability for any given gene. There's huge variability across cancer types on its relative frequency. And then the other thing to observe is that as soon as you get past this main gene TP53, the relative frequencies are extremely low. And then it gets even worse. So if you go, that's the top 20 genes, and if you go to 100, then you start seeing this really strong, heavy-tailed effect. And then this tail continues. So if you go to 1,000 genes, it's a really, really flat distribution. And why is this important? Well, imagine there's a patient and it has a relatively rare mutation over here, but this has the same downstream phenotypic effect as one of the more well-studied high-frequency genes. And so if you don't know this, then you will not necessarily find this in your typical clinical setting. So somehow you want to learn from the data that these genes should be combined together to in one kind of disease state. So this is the sort of state space view of this this is sort of like a variational auto-encoder view of things. So we have this RNA space in a patient here. We're going to map this to a latent space such that if we sample from in this area here and go back to the RNA space, we have good reconstruction. So we want a latent space that has good reconstruction for our RNA. What's different now is that we are including the DNA here. So we're saying that the mutations for a patient should map to the same location in the latent space. So this is reconciling the DNA and RNA here. And this is one of the reasons why we want to do this is you could, like I said before, you could have the, 2 different patients, the star and this cross, that have the same RNA profile but have very different DNA mutations. And what we want to learn is we want to sort of learn a structured prior mapping from DNA to the latents that sort of captures that these 2 genes should influence together this single factor. And then there's very few equations. This is just the variational lower bound, and just putting into equations sort of the intuition that we have, that we want to learn a latent representation Z that has good reconstruction. But at the same time, we want to make the distributions conditioned on whether you have your mutations or whether you have your RNA to be the same. So that sort of minimizes this sort of distance here. Now, the structured prior, I haven't really spoken about what it actually is, and this is sort of, as this is work in progress, it's just like sort of a first version of what it is. But the high-level idea is this. So each gene has a set of weights that go into the different factors. So there's k weights per gene, and they're constrained to sum to 1. So these genes sort of have to decide which factor it's going to influence, and you can encourage it to have, by engineering the sort of the entropy of this distribution, you can say, well, you should be, very strongly influence one or the other. And the other aspect of this is that it sort of, you can also force genes to make a decision. So if these low frequency genes, you can say, well, we want you to influence some factor, so you should decide which one it is. And so this way, all the genes sort of have to make some decision on which factors they are influencing. And then what this summation does is it basically goes through all the genes, and it says, okay, if you have a mutation and you're an influencer for this, then we'll turn on this factor. And you can imagine that if you have mutually exclusive genes in your data, that a low frequency and a high frequency one, that they'll both be influencers. And in the rare mutation case, they'll also turn on this factor the same as with this the high frequency mutations. And there's other parameters that sort of say, well, we don't know how many mutations should turn on this factor and so on. But the main idea is that mutations should decide which factors they influence, and then all mutations should decide on some factor to influence. So the data, this is just the colorectal cancer data. On the left, you have the DNA mutations just binarized, and you can see the heavy tailness and sparsity of the data. This is just the top 100 genes. And on the right, we have the RNA data, and you can see there's a lot of structure in this data as well. So we are mapping DNA to this discrete latent space, again, to the RNA. We're not so interested in this. This is the sanity check. The reconstruction works quite well. But what we're really interested in finding out is what is being learned in this structured prior. And so here I'm plotting sort of the influence, the main influence for each gene, and they're kind of tied together. So these circles are sort of showing that these sets of genes are influencing the same factor. And what we want to do is then inspect, functionally, are these doing the same thing, and is our model making sense? Okay, so the future work is mainly to sort of continue and sort of validate that it's actually working and finding this structure that's sort of matching the intuition that we have. And I think the most interesting is to expand to pan-cancer cohorts. And there you can really leverage these different mechanisms that the different diseases have. And sort of as Danielle said, in clinical trials, what typically happens is you put all your cancer patients with the same type in the same trial, and then if you have a drug that's targeting some particular mutation or some particular phenotype, that drug can fail because only a few patients in that study respond well. Whereas if you can classify your patients according to the disease state that you've learned in your model, then you could perhaps have all the patients from that with that sort of that genotypic sort of disruption, that disease factor in the same cohort, in the same trial, and then that drug would then be quite successful. And of course, by learning this across pan-cancer, you can do better personalized treatments by grabbing treatments from a small cohort that has a drug that works on some one particular genotype, and then use that in other across to the other cohorts by using these disease states. Okay, thank you for your time.
We have time for questions, but can I ask Volkan's already set up any questions?
I guess one tricky bit here is that there is like a between genes and RNA there's like a one-to-many mapping, because a single gene can give rise to different types of RNA. Is your model taking that into consideration? Or does it matter at all? I'm not sure.
The genes mapping to multiple RNA?
Yeah. There's a mechanism called splicing.
No, that's not taken into account at all. In fact, I don't think there's not really any way of having that in the data in the first place. It's not at that granularity.
Okay.
Yeah.
That's a really good talk, Ted. Thanks. Could you explain why we need a latent variable model here? So we're sort of learning, in one sense we could view this as just learning a discriminative model that goes from DNA to RNA. And we could just directly parameterize that mapping without latent variables, perhaps with a bottleneck in the middle with a sort of a low-dimensional representation. Is it clear that we need the VAE and sort of actual latent variables in there?
Well, I guess in that case, I mean, if you look at the objective function, the disease is that can be thought of that bottleneck or a hidden. So you're basically going DNA to bottleneck to RNA, and most of that is the same, maybe objective function that you're thinking of, except that you have this extra one where the RNA also has to map to the same disease, the latent space. The reason why you want this is, one of the reasons is that a large part of the signal in the RNA will be sort of what the cells are doing because they're that kind of cell, and not necessarily what the disease part is doing. And so, I mean, I'm not, yeah, I think there's lots of different ways of doing it, but one of the reasons is that you want to capture just the disease part in the latent spaces and not the sort of the RNA part, and you may, or the rest of the cell, normal cell functioning part, but there's not a lot. This is just like one modeling choice, really. That's what it comes down to. Yeah.
Sounds like a great